Hello friends. In the last video we had talked about different kind of DNA polymerase and the initiation complex in the eukaryotes and the sequences in the replication origin in case of eukaryotes. And now what I'll be discussing is on about polymerase switching. This is called polymerase switching. As I had already stated that the DNA alpha DNA polymerase alpha functions similar to the primase, and it synthesizes the RNA template on which the DNA polymerase delta or eta functions, because they cannot start the DNA synthesis on its own. It needs a RNA primer on which they can add up some more nucleotide to elongate the DNA strand. Now this DNA polymerase alpha, which initiates the replication after synthesis of 20 to 30 bp of this RNA template, it eventually falls off, and uh, it is replaced by other polymerase such as delta or eta. Delta is for the lagging strand and the eta is for the leading strand. So in case of alpha, it is replaced somewhere, and is replaced by delta or eta depending on the which strand they are synthesizing, and this phenomenon is called as polymerase switching. Now, each and every DNA polymerase has a specific characteristics which we call as the processivity. This is called as processivity. Now, what is this processivity? It is nothing but the how many numbers of nucleotide are added per binding between the DNA polymerase and the DNA template. As we know that this binding of if this is the DNA, and the binding of the DNA polymerase, it then starts to put the nucleotide in it, and as we know, the equation of the elongation of the replication is DNMP. This NMP refers the nucleotide monophosphate. Suppose it has n numbers of monophosphate, and then DNTP, that is nucleotide triphosphate, is added, and then the reaction product will be DNMPN plus 1 and plus PPI. We know that nucleotide triphosphate has got 3 phosphate group and 2 phosphate is removed, so it forms dinucleotide. It forms the nucleotide monophosphate, and it is added with the already present deoxynucleotide monophosphate polymerized string, and the PPI represents the 2 dissociated phosphate group from the NTP. So now that how many uh, how many numbers of nucleotide are added by binding event between this DNA polymerase, means the number of nucleotide, the more number of the nucleotide is added per binding of the RNA of the DNA polymerase with the DNA, is actually directly proportional with the processivity. That means the processivity increases with the number of added DNTP. So the more dntp added, the more processivity. And the non-processive dna polymerase are actually those that that adds fewer number of nucleotide per dna binding event. So processivity is actually the functionality and how fast it is being able to do this. They're talking about this enzyme. All right. So now another important is the rate of the reaction, that how fast this replication fork is moving. So the rate of the reaction is how fast this replication fork is moving and how many base pairs are getting added per second. So in that case, this in case of this prokaryotes, this reaction rate is much higher, as high as 1000 base pair per second per fork is the speed, 1000 base pair. And in case of eukaryotes, this is 10 times slower, and only 100 base pair is added per second per fork. Now the question can come that why this fork movement is much slower in eukaryotes than the prokaryotes. Then the answer is this, that this DNA are coiled around the histone proteins in case of this eukaryotes, whereas this in this case of prokaryotes there is no histone molecule, and this DNA molecule is more or less very much pure and without any contamination of the protein. But in case of eukaryotes, the DNA is tightly coiled around the nucleosome, which is made up of histone proteins. So first uncoiling this DNA molecule from the from this nucleosome, and then to replicate that DNA, this process takes a little bit of time. So for that reason the fork movement cannot be that much faster. Okay. So now we can go we have come at the end of this replication and everything. And before this I go to the reaction, the terminal reaction of the replication. I would like to talk about the removal of the RNA primers. If we remember that this was the replication fork, and this was the SSB, single stranded binding protein, bound here on the single stranded DNAs, preventing them to recoil. And this is my helicase enzyme running towards this side and producing supercoiling. This is the place where supercoiling are getting generated, and the topoisomerase 1 enzyme removes this supercoiling. And here another thing happens, that is let me take another color. Okay, this much is fine. That RNA primer is synthesized. RNA primer is synthesized here, here in case of this lagging strand, and new DNA molecule is added on it. Sorry, my line is not it's okay. Now the DNA molecule is added on it by the enzyme called DNA polymerase. In case of this leading strand, this the eta polymerase is responsible for synthesizing the leading strand. In case of this lagging strand, small segment of DNA molecule are synthesized by the delta, this DNA polymerase enzyme, and the small fragments are called Okazaki fragments. Now look at this, there are small RNA templates are there, which actually functions as the start site of this DNA replication, but we cannot keep it keep these RNA structures within the DNA structure. So for that reason this RNA has to be removed. Now how this RNAs can be removed, here comes another protein which is known as Rnase H. Rnase H, and this chew up these RNA fragments. It chew up this RNA, small RNA segments, and it removes the RNA. But the problem is that Rnase H cannot cleave the bond between the first nucleotide of the DNA and the last nucleotide of the RNA. So that is removed by the enzyme called FEN1, and this FEN enzyme one, which is known as flap endonuclease 1. This one actually breaks the bonding between the last RNA nucleotide and the DNA nucleotide, means the last RNA nucleotide which remains bound to the first DNA nucleotide by the phosphodiester bond. This bond cannot be cleaved by RNase H. Rather, this is done by FEN1, this enzyme. So in this case, the RNA molecule is removed, and these small gaps are now filled up with the DNA pol 1 in case of the eukaryote, in case of these prokaryotes, and in case of this eukaryotes, there are a small gap filling polymerase are involved, which fills up the gap, and now the complete structure of the DNA molecule can be seen, because these RNA are removed by the Rnase H, and in case of prokaryotes, it's synthesized. This gap is filled up by the DNA pol 1, and eukaryotes have different kind of enzymes. Now in case of these Okazaki fragments, there are small these Okazaki fragments are still not joined together, and this joining is done by the enzyme called ligase. Ligase is the enzyme which ligates the Okazaki fragments with each other, and so we can get now here a continuous DNA strand like the leading strand. Okay, so I'm not really good at drawing, as you know already, but this is the leading strand which was synthesized continuously by the DNA pol 3 in case of prokaryotes, and this lagging strand was also synthesized by the DNA pol 3 in case of these prokaryotes, and in case of eukaryotes it was synthesized by eta, and it was synthesized by delta. This lagging strand was synthesized with the Okazaki fragments, and then the RNA was removed by the Rnase H, and the last RNA DNA nucleotide bonding, this phosphodiester bonding, was broken by this FEN1 enzyme, and then these small gaps were filled up with the DNA polymerase 1. So I guess this much of information was fine for this video up to now, and I'll see you in the next video. Thank you for watching. Bye.